Hi everyone, good morning and welcome to week 3 of your journey into the world of the unseen. So the big takeaway from this week is that, bacteria are sneaky little boogers. Okay, I know that is not exactly proper scientific verbiage, but the fact is, bacteria are, for the lack of a better phrase, sneaky little boogers. Now, over decades since the development of the very first antibiotic, you know this as penicillin, that was back in 1941, dozens of others have been developed, put on the market, and are currently in use in medical settings around the world. And yet, as brilliant as human science and medical researchers and engineers are, those sneaky little boogers, the bacteria, still seem to devise ways of skirting around our humans, attempts to control them, destroy them, and minimize their damaging effects upon us. And once again, the most unsophisticated organisms on the planet seem to have the upper hand over the most sophisticated organisms on the planet, which makes me wonder once again who really is in charge down here. Well, down below I've provided a link to a short video from Science News, and in it you will see how bacteria are able to develop um, resistance to antibiotics and confer that resistance onto their progeny in very short order. And so when you recall that the generation time of Escherichia coli is only about 20 minutes, you can appreciate how fast things can happen in the world of bacteria. And as you culture bacteria in this week's exercises, pay particular attention to how fast those sneaky little boogers can do their work. So this week you'll learn to culture bacteria using aseptic techniques. You're working on the streak plate exercise and the Kirby-Bauer assay exercise. And you'll learn about the effectiveness of various antibiotics, antiseptics, and disinfectants in controlling or inhibiting bacterial growth and reproduction. Housekeeping items for the week. You should have your microbiology kit by now. I haven't heard from anyone, so I think everyone has their kit. Um, and if you haven't started on the exercises already, which I kind of gave you a heads up over the weekend, you definitely want to get started on them today because because your cultures, you initially rehydrate your cultures. It takes a couple of days, about 48 hours, um, at least 24, for them to incubate. You'll have your tube and the liquid broth inside will be clear after you add the bacterial culture. It's going to be clear. And then after 24 hours, you should be able to look at it and it's starting to get cloudy. That cloudiness is a good thing. That means there are bacteria cultures growing inside. That's what you want to see. Okay, more on that later. Make your initial discussion post no later than Friday. That's July 4, 11:59 p.m. GCU time, and your peer responses no later than Sunday, July 6, 11:59 p.m. So, I know Friday is July 4, and I know that this weekend is a holiday weekend. All right, that's enough to be said about that, okay? If you have family plans for Friday and for the weekend, then you want to make sure you get your stuff done earlier in the week, because there is no extension, for example, on your initial post past Friday. So if you're planning on doing stuff for the holiday weekend, you need to get your initial post and other things done earlier in the week. Okay. And unfortunately, the practical exercises, the streak plate and Kirby-Bauer that you're doing, unfortunately, that's going to go through this week, and everything is due on Sunday, and it's a holiday weekend. I get it, all right? You're just going to have to work around that as you best see fit in your circumstance. All right, we're working on topic 3, methods of culture transfer, pure culture isolation, antimicrobial testing. This is T3 or topic 3. Complete the pre-lab questions for 3 and 11. They are due no later than Sunday, July 6, 11:59 p.m. Start working on your lab activities today if you haven't already. There are 2 of them. These cultures need time to incubate, at least 24 hours. Usually, since you don't have an incubator at home, usually it's going to take 48 hours. When you incubate these cultures, you want to put them in a warm, dark place. All right? Remember, optimum conditions for bacterial growth are warm, moist, and dark. The moisture comes from the medium, from the nutrient broth. So you don't have to worry about that, but you still need to put your cultures, put your tubes in your little plastic rack, and you want to, and by the way, I posted a video on how to reconstitute your cultures, and I only did 2 of them. Make sure you do all 4 of them at the same time. Put your tubes in your little rack, and put that in a warm, dark place. I don't know what it is and how it is in your house, but don't put them in the refrigerator. Don't put them next to the AC or the swamp cooler register or whatever. Some students put them on the shelf in their closet. I used to put mine on top of the refrigerator because it's dark up there and you get the heat from the refrigerator providing the heat. But the first time, a couple years ago, when I first started teaching this class, actually 2022 is my first time, started teaching this lab, and I put my cultures on the top of the refrigerator and they didn't do anything. So what I ended up doing is putting them in a box, just a cardboard box, and putting a desk lamp over the box. So the darkness, they're in the box so that it's dark and the lamp is, shining down in the box. That provides the heat. So however you want to do it, you don't want to put the lamp directly onto the tubes, right? They need to be covered up. So again, they need to be dark, they need to be warm. Don't put them in the microwave. Um, and they need to be moist, but you get that from the nutrient broth. And then after you inoculate your agar plates, after 24 48 hours, which is when you're going to do your streak plate that's on the agar dish, and your Kirby Bauer, then same thing. The moisture comes from the medium, the agar, and you need to put those plates in a warm, dark place in order to incubate. Okay, more about that as we go through the week, but kind of gives you a heads up. But if you want to know, I posted a few videos on how to reconstitute your bacteria and also streak plate, quadrant method for streak plate, and also the Kirby Bauer. And also, when you go to the instruction manual for these exercises, you'll find those on the right side of your topic 3 page. The right side, there's a vertical menu. Scroll down the vertical menu. Down near the bottom, you'll see instruction manual or lab manual for the streak plate. I think it says streak plate isolation method and the Kirby Bauer assay from Carolina Biological Supply. Those are the 2 manuals that you want to look at for these exercises. Someone posted a while back an instruction packet from GCU. Don't look at that one. You look at the ones from Carolina Biological Supply. All the instructions are in there, and I think they're relatively easy to follow. But certainly, if you have questions along the way, let me know. Okay. You're going to use your cell phone camera to record your results, right? So you need to take pictures, photos of 24 hours, 48 hours, or whatever it says on the data sheet. and it's those photos that you're going to upload or attach to, just copy and post them in your data sheet, and that's what you're going to turn in for the streak plate, and then you're going to post your photos and also complete the data sheet for the Kirby Bauer. Okay, again, more on that later, but just use your cell phone to take those photos, no problem. Okay, and both of those assignments are due no later than Sunday, July 6th, 11:59 p.m. There's a little bit of give time there because sometimes it takes a little while, depending on where you are and what your atmospheric conditions are. It may take a little while for your cultures to incubate, to actually grow and show significant growth so you can complete the exercises. So I'm a little variable there, but do your best and we won't have any issues. I can't wait to see your results. I always look forward to seeing student results here. And it's just fun, and it's great for you to be able to see bacteria growth. You can see it. And by the way, yes, you can smell it too, because bacteria, when they're growing, um, well, they have a very noticeable odor, which you will see. Okay. Questions, issues, problems, contact me as soon as possible. Message 804-229-7993. Use your Halo message box. Two quickest ways to get a hold of me, and certainly campus email. I posted a short video about the evolution of antibiotic resistance from Science News. It shows you how bacteria can grow, how quickly they can grow, and pass antibiotic resistance onto their progeny. It's a short video. You can kind of see in real time what's going on there. It's very 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 quickly. I think it should be eye opening for you. All right, that's all I have. I'll be back with you as the week goes on. Let me know if you have any issues with the exercises, if you have any questions. And that's that. Have a good rest of the week.